Hi, friends of the sun. Welcome to our weekly reading group happening every Monday, and you can join the Zoom yourselves, ask authors and presenters questions. And yeah, the thing that we usually have going on here is that someone writes a nice paper. I find a nice paper. I think it's interesting. I want to talk with them about it. I ask them if they want to present it in a reading group, and then we have yeah, discussion about it. And you can join these discussions yourselves and ask questions to these authors. And yeah, happening every Monday. Hope you join. All the information is in the description. And with that, today we're talking about some genotype to phenotype mapping stuff with Farhan. So let's
We
go.
have different types of cell type. We know different tissues and all of these things, and that's basically what boils down to, which is the genotype and how it emerges to become phenotypes. But also in the context of pathology, we know a lot of diseases are related to that, and any therapy that we design, it's going to be basically targeting those gene relationships to kind of fix the phenotypes that we expect. So understanding this phenotype and genotype relationship is very important to understand what is the decision-making process in cells. And that could be really important for basically understanding biological phenomena and also uh, maybe hopefully designing therapeutics. I want to go back to, what genotype and phenotypes are. And my favorite figure in the history of genotype and phenotype is Wilhelm Johansen. He was in 1909. He published a book in German, which has not been translated still in English. But he first introduced the concept of gene, um, which basically you can see here. He got the concept from pangene that Darwin introduces, and he says that it's better to say gene because it's free of any connotation or hypothesis. And then he goes on and introduces the concept of genotype and how gene interactions become important. He also introduces the concept of phenotype, and he says that phenotypes are important because that's how genotypes become realities and how they basically manifest. And phenotype comes from basically the phenome. It's a Greek word that shows just appearance, whatever we can see or whatever we can basically measure. So in modern genetics or modern synthesis, we understand genotype phenotype relationship in two different ways. Either we have a phenotype and we try to understand what are the genetic backgrounds of that phenotype, which we call forward genetics, or we have a genotype and change genes here, and we want to understand what kind of phenotype emerges. This is very common nowadays. Any type of study that's happening in genetics or molecular biology is basically one of these uh, forward or reverse genetics. In the context of disease, we get samples from patients and we sequence it to understand what are the genes that are important. Or in the context of synthetic biology or molecular biology or perturbation analysis, we go and we'll just perturb different genes and we play around with them to understand what kind of phenotype emerges. And there are some challenges with like studying genotype phenotype relationship this way because we know that the genotype landscape is huge, right? If you want to perturb each single gene for one, for example, human cells, we're going to do like 20K single perturbations. If you want to study double gene interactions, we're going to do like 200 million. And if you want to like do 3 genes at the same time, we're going to do 1.3 billion, just going to exponentially increase. And this is not something that is basically doable with the technology that we have right now. And another important point that people usually forget is that genes are not simply just being expressed. They're not just switches that just become on and off. They are on or they become active for a reason. There is a context that makes them activated. It's not that there is a CRISPR Cas that sits there and just like deactivated. They're just like simulating it like that. Maybe is not the best way of studying genotypes. On the other hand, phenotypes are also massive. The landscape of phenotype that we see is huge. We have different species in multicellular organisms, we have different cell types, we have different tissues. In human, we have like a huge amount of diversity in phenotypes that we see between the species and different cell types. And just conceptually, I like what Edmund Husserl says about phenomenology, that conceptually phenotypes are subjective. You know, we can't really say a phenotype is, we can't really measure a phenotype in a way that's very qualitative. So there are certain challenges that, like what Husserl says, like our consciousness also plays a role here. So these kind of challenges made me rethink how we approach phenotype-genotype relationship, and maybe create a new way of doing it. So what I propose during this paper is that we can study genotype-phenotype relationship simultaneously. We can basically perturb genes and see phenotypes, and we can perturb phenotypes and study genotypes at the same time. But this is really hard to do because, you know, genotypes are high dimensional space, phenotypes are high dimensional, they're contextualized, they're dependent on the organism that we are studying. So it's not really easy to do that. We have to, you know, rely on computation at some part of it. I argued that, you know, with the technology that we have right now, for example, single cell RNA sequencing, and with the language models that are becoming more popular, it's becoming easier and easier to study this kind of relationships. And that's what basically I'm proposing here. So we're trying to study genotype-phenotype relationships simultaneously, and we encode genotypes and phenotypes in high dimensional vectors, and we try to perturb them at the same time. And understanding this relationship is possible through language models. So I'm going to go through what are the I'm going to skip this one. Like I said, it's like high dimensional, and but yeah, I'm going to go through this in a little bit. But the way that I think about it is, you know, masking in language models. We have some phenotypes, we have some genotypes, we are masking them, and we want to understand what is the relationship between them. So in this study, I used BERT as a bidirectional context-dependent prediction transformer architecture, and pre-trained it on a lot of, you know, single cell RNA data. And I'm going to go through how this will be possible with like some mass language modeling. Basically, this is how we have designed the model. So we have genotypes, which are single cell RNA data from basically RNA expressions. And we have phenotypes that are basically categorical information or text-based information for this kind of genotypes. For example, we usually get we have a lot of single cell RNA data. We know that which sex they're coming from, what's the age of that donor, what's the tissue that's coming from. Most of the time, we've manually annotated the cell type. Sometimes it's not clear, but there are other things like we know what disease it's coming from. We know what kind of, you know, ethnicity they have. All of this information is like basically phenotypes that we can encode to the model. So we trained it on Tabule Sapiens, which is just a data set of 500,000 cell types from 14 donors, 25 different organs, 400 different cell types, 12 age stages, and you can kind of see a distribution of cell tissues for each donor here. And the way that we encoded them is like basically jointly training them, jointly training phenotype and genotype at the same time. So you can see like phenotypes are categorical here, and genotypes are basically gene expressions. And we have a vector of cell state or a state that has been encoded with phenotypes information like sex and age, and genotype information like expression of each gene. And then we assign like vectors like just normal language modeling here, and we embed all this information. It goes through an architecture, and, you know, some of this information gets masked here. For example, in genotypes, we get like 15 to 20% of them get masked. In phenotypes more, because we have a smaller amount of phenotypes here. But the way that the model learns is that, okay, like there is a female information coming. I don't know this phenotype supposed to be age. I don't know what age it is, but I know it's from aorta. I know like a bunch of other information, and I know this gene exists, but 15% of the genes don't exist. So as the model gets trained, it understand, okay, there is a relationship between genes, that's the gene-gene interaction, and there is a relationship between genes and phenotypes, basically. And the output of the model is just, embeddings that are information rich and they're contextualized, and they've learned something about not only the gene interactions, but also the relationships with the phenotype. And the same thing happens for phenotypes. For example, the aorta has been encoded with a lot of information about the genes that have been active in that, but also about general observations of the phenotype, like what kind of age, what kind of sex, what kind of cell types exist in the aorta, and so on. So I'm going to just present some very a small amount of data here, just show that this kind of encoding of information can improve how we understand genotype phenotype relationships, basically. So just looking at the confusion matrix, I forgot to say here, basically we're doing binning for each gene expression. So each gene gets a number, which is the bin number, and also a position. A position is always fixed for a gene, and all of these things have fixed positions. So you can imagine that this is just a network that's been updated. But unlike language that we have, the positions are important to, because we're predicting positional relationship. Here, positions are not changing, and we have a fixed position for each gene because the data is permutable, right? We can change gene 1 and gene 2, their position, and nothing should basically happen, which is not the case in language, right? So a lot of people have actually used single cell RNA in language models for single cell RNA. They have made this mistake. There we have 2 models already published, GeneFormer and single cell GPT. Both of them, they have considered it the position, which is basically wrong. So they have done some tricks and everything, but this is not the way that we can encode information, at least in genetics, that we have permutation-free variants. Looking at confusion matrix, you can see yes.
Can we go a slide back and just step away a little bit, and maybe it's also good to summarize for the rest of the people. So what we do, right in the end, you want to map some genes in the forms of expression data. Is that right? Or is it genes in the forms of DNA?
Yeah, in form of expression. Yeah.
Okay, so we then have G. So we have for each gene whether or not it's turned on. Is that what the 1 and 0 should indicate in your figure?
Yeah, so the first step that we did was just binary modeling, right? If a gene is on and off. So we kind of define a threshold for gene expression. We said, okay, if it's lower than this, it's on, and it's higher than that, it's on, lower is off.
Okay.
We tried the model binary first, but at the end we did binning, right? So the expression just got binned into special buckets. I should update this figure, just show that it's not only 0 and 1. There's 5, 6 other numbers here.
It's I don't know, 100 categories per genotype, indicating how much it's expressed or something like that. And then we have also just a text document, or where does the text come from, basically?
Yeah, I think I said text-based. This is mostly categorical information, right? We have sex, for example. We have this cell comes from a female or a male. Then we have age, what kind of age this gene expression is coming from. 10 years old, 20 years old. It is possible to encode others. So we've done that, other type of modalities here. You can basically any type of information that's coming from this experiment, you can encode here. You can design other vectors here and add other text-based information to it. Or something that we talk, and it's you can basically encode information about the protein structure here, which I think is our next step that we're thinking. But that's also an interesting route to go.
Okay.
Yeah.
Then we also have in the I think this is somewhat related to a question in the chat. Like, why do you want to and right now you train this with masked language modeling with like masking what percentage of the tokens?
Yeah, so for genotypes, we did around, I think, 18%. That was the ideal, the optimized number. And for phenotypes, we did 50%. So they different
Optimized for
masking
what?
ratios.
For ability to predict
For performance.
phenotype?
Yeah, for genotype. So firstly, yeah, and I quickly see.
Why genotype? I mean, in the end, aren't we interested in predicting phenotype?
We are also interested in genotypes, right? Like given that we have a tissue or like some information about a phenotype, we want to know what genes are being expressed and what are the ones that are maybe more important than the other ones.
So we basically, the task is to in-paint the missing information. Like whenever you have some phenotype data, some genotype data, we want to in-paint all the rest.
Yeah, exactly. I think that's basically what we do in perturbation analysis, right? We perturb something and want to see what happens at the end. Here we're doing the same thing, but a little bit more maybe what I call like integrated method. Like we both have phenotype and genotype because we don't have counterfactual information. We don't know if this gene. I mean, if we give it a lot of information, we probably the model is going to learn if this gene is on or off, how it's going to change a phenotype in a hypothetical situation. But for now, what the model is learning is basically how their relationship is encoded basically in this information.
Yeah, sounds good.
Yeah, so just confusion matrix, normal stuff. The model can learn really well, like age, tissue, cell types. Like these are huge categories in the cell type. We have 400. Sex is pretty easy. The binning has been a problem. It's the lower like performance wasn't finding the binning number, and we have some ideas here. We're trying to like do dynamic binning and all of these things because genes, when we have a gene that's being expressed from 0 to 10, it doesn't mean that it has the same effect if it's in the range of 0 to 5 or 5 to 10. The relationship is not linear, so we're trying to make it dynamic, so the model can learn what is their best range for each gene and what is the best bucket for binning for each gene.
How do you evaluate here? Say you, I want to now predict, let's say, I don't know, what is a good phenotype?
Anything you can say, like tissue, I don't know, or cell type.
Okay, cell type. And then, so I want to predict cell type, then what is the information that you provide as input? Like all the genes or maybe half of the genes or just according to your masking ratio?
Yeah, so you can it depends on the task, right? Like I have defined like different tasks for this paper, but you can also define tasks as like, okay, I have a phenotype and I have like 5 genes, and I want to know what are the rest of these genes being expressed. Like that's also a task that you can define. But this is like how the model is learning. That's what the model is maybe good at. I don't know if you increase the masking ratio, what happens? That's a good question. That like I don't know if you give it like 5 genes and 2 phenotypes, what's gonna spit out? I haven't done anything like that.
No, but I mean, so what is, we have here some confusion matrices, or we have some diagonal lines, basically, is what we
Yeah.
can see.
Yeah.
Let's maybe look at the numbers down there. And like, what are you predicting there? And like, say, if I have the sex as input, as the phenotype, and okay, this is an example, but say I have the sex as input and the number the number of X chromosomes, whatever, as input, then or no, I want to predict the sex and I have the number of X chromosomes as input. Sure, number of X chromosomes is not a phenotype, but yeah, then this task wouldn't make sense. So what is an example of a task here where
Yeah,
you're predicting a phenotype, and then what are the other phenotypes that you have as input?
yeah. Let's think about age here, right? Like I have all the, let's say I have everything in my model except age, right? So the age that is come with the data, the label that's coming is like 44 years old. And I want the model to predict what that age is basically at the end, right? So that's what the confusion matrix is showing here. So I've masked the age and I want to reconstruct it on the other side
Okay.
based on gene expression
And
information.
what is then basically the most similar or the most correlated feature or the most correlated phenotype with age? Like
Uh,
what would be the
with age,
confusion matrix between age um and phenotype 2 and phenotype 3, and then you have all these confusion matrices and you see yeah, which how much better is your model, or how much or is this basically trivial to extract the age
yeah,
from the other?
I think age is really hard actually to extract from, like uh age is something that um is difficult to understand from gene expression data. That's something some of the ones that are really easy that I have more data about this, but it's, cell type, if you do some normal clustering on your gene expression data, you can kind of understand what cell types belong to where. But age is not easy. Age is like really dependent on really more. It's not only one gene it goes up and one gene comes down that shows like this is what age. There is like more intricate like relationship between gene expression. That's the hope that we're trying to
Yeah, but I
understand.
mean, you don't only have gene expression as input, right?
Yeah, yeah, but everything else, you mean the phenotypes, right? Like for example, we have like, let's just forget about genes. Like we have sex, right? We know that there's no correlation between sex and age. At least in this data set, everything uh, has been balanced. Basically, the data.
But if we had like number of gray hairs as input as phenotype as well, which probably you
Yeah,
don't,
yeah,
but
yeah,
I don't know
definitely.
what other phenotypes are.
Yeah, the phenotypes are age, sex, tissue type, and cell type.
Oh,
So
okay,
these are
so
they
we
have
only have
yeah, yeah.
this.
So they have no correlation with age. They are a correlation between tissue type and cell type. That is true. And sometimes between sex and tissue type. There's some tissue types that only exist in one sex and the other ones. But even for tissues that exist in both sexes, it's really hard to predict which one belongs to what sex.
Yeah, and by the way, we can't read, or at least I can't read what's on these figures.
Yeah, I need to make it like a little bit bigger. Yeah, I wanted to just condense all the information here.
Yeah,
But let's just move on maybe on this, and we can I think it'll become more clear uh what kind of like predictions we can do. This is like basically looking at the raw data, right? Like just doing a UMAP on the single cell data for like that 10,000 shot like random samples from the data set. And that's what you get usually, like when you see like a lot of people doing that in the single cell field is like if we do clustering, it'll be like, okay, these clusters show whatever gene, this cluster do whatever gene. But when you feed it into the model, and you can see that the clusters like really get separated, which is is what we are looking for at the end of the day, right? Like here we have like a lot of things just clumped together. We don't know what gene, what tissue is here. But here we can see like everything is separated. I know that the colors, I mean, I can't really find 40 colors that can, maybe there are better ways of showing it, but we'll try to, label them here. But even the tissues that are more similar to each other, the model can learn that like these are the tissues that are more similar and these are the ones that are more different, based on the gene expression that's been fed into the model. Same thing you can do with age and sex. Like this is basically just doing a PCA on sex from the model, and this is what you get when you take under raw data, right? And same thing with age. Age, I mentioned that age is really hard to predict because it's really hard to understand aging process from gene expression data sets. We have like a lot of other things involved, but the hope is that we can, extract as much as information as possible from gene expression data by encoding a lot of more information into it. The other interesting thing is like when you look at every phenotype separately, right? Like for example, for tissue, as I said, like the model can learn that there are similarities, right? Like between vascular cells, they all cluster together. The muscle cells, they're all similar to each other. The skin cells cluster together. Pancreatic tissues, cardiac tissue, and lymphatic tissues. So these are the things that we know, and the model also can recognize. But there are some anomalies here. Like for example, tongue and kidney and retinal neuronal layer, they cluster together, and the model identifies them as similar. I did more digging into it, and I presented more stuff in my paper, but it turned out that there are actually a lot of similarities for diagnosis between tongue and kidney. A lot of studies that have been published, and it's really new research that shows that chronic kidney disease associates with tissue or the tongue abnormalities, right? The association is really high actually, and there are like people right now trying to understand why this relationship exists, and based on color and texture of the tongue, you can understand if the kidney is healthy or in a chronic condition or it's failing condition. So these are just cross tissue understandings that you can make that usually you can't really do with other methods. And the other thing is, we know like gene interaction networks are scale free. You can also extract it here. Just looking at macrophages and stem cells, you can see that there is like scale free relationship. Scale free networks we know that protein interactions are scale free, and people have shown that. But for gene networks, it's been harder to do. But basically, we can do it here with looking at embeddings. And the interesting thing is like based on only this gene interaction, you can extract a hierarchical cluster of how genes work together. So I would like to, maybe propose this as an alternative to, pathway analysis. Pathways are usually very simplistic way of looking at this gene interacts with this one and that one interacts with this one. They create this pathway, but it's better to look at them as a network that they are interacting with each other and they are affecting each other in some ways.
So can I summarize or understand a little bit here again? You take your embeddings of individual, like the tokens of corresponding to this gene at maybe the last layer of your transformer or something like that.
Exactly.
And then
Yeah.
you cluster all these embeddings. Is that right?
Yeah.
And how do you cluster them?
So this is just based on similarity, like Euclidean similarity. I just find similarity between genes and
Yeah, but how do you create it? Do you use some clustering algorithm?
yeah, I used Tisney basically for this one, I think. I forgot exactly which one was it, but I think it was Tisney.
Okay, but with Tisney, you wouldn't create clusters, right? With Tisney, you would just reduce the dimensionality in a similarity preserving way.
Yeah, I think I forgot exactly which one was it, but I can yeah, if I can
No,
dig
not
into it.
so.
I forgot it.
Maybe it's just some k-means clustering or something like that. And now, how do you propose to use this to replace pathways? Or is that
Yeah.
what?
So yeah, so I talked more in about this in the paper, but I should have brought more information about this. I thought this would be interesting. But basically, like we know that the genes work, they interact with each other in the molecular level, and these interactions like create some type of new, like two proteins come together, genes interact with each other and then create a new form, and that form has a function and create like interacts with other molecular interactions. So this kind of hierarchical interactions can be, I think, is better way of looking at gene interactions, or like analysis of gene network, rather than just pathways. That pathways are, we are trying to just create one line of, okay, this protein interacts with this one, that one with that one. We're just following one protein, whereas these things happen at the same time.
Okay, so the purpose of this pathways is to let me see, is there someone? No. The purpose of these pathways is to kind of find out um how hmm, or like which genes interact with each other. Is that right?
Yeah, yeah.
Yeah, now
We can we
you're
can get we can do the same thing here, right? Like we can see what genes are interacting, but we can also see what is the cluster of genes that this interaction is happening in, and which ones are interacting directly and which one are secondary interaction and tertiary interactions, right? Um, so, but
okay.
it becomes more interesting when you look at it in the context of a phenotype. I looked at it in the endothelial cells, like young endothelial cells and old endothelial cells, and you can see the scale free pattern is pretty much there, but like there are the nodes that we call like hop nodes, the nodes that have like a lot of connections. They kind of disappear, like they lose some of their connection, and it becomes more clear when you do this kind of clustering, right? You can see, I can just label them like these are specialized, I call them specialist genes. These are genes that are specifically important for endothelial cell function, let's say adhesion molecules, and the ones that are activated during coagulation and immune related genes. You can see these are the young endothelial and these are the old endothelial cells. And as the cells become older, they lose some of their specialized genes, right? Or they like kind of like the specialized gene becomes more generalist, basically. And that's just losing some of the connections that they used to have. And this is like an interesting way of looking at it. And I talked about what a specific genes are losing their identity during this process for endothelial cells. But basically, this happens for a lot of different cell types and a lot of different tissues. And that's like why we get this, high level sort of characteristics of aging that's really hard to hone it down to one or two genes. Uh, yeah, so I'm gonna skip this, but basically you can, just looking at the gene interactions, you can understand the same genes, how they interact differently within two different phenotypes. Let's say here, macrophages and parasites. So you're mute, I think.
Sorry about that. Just some questions from the chat. Have you tried to experimentally validate some gene interactions that you predict?
Uh, no, we haven't done any type of validation yet, and that's the next step. Like this was more like the method development for this work, but we're doing some validation specifically on the endothelial cells, because our lab is mostly vascular cell lab, so we're interested in this kind of work.
How are the specialist genes identified?
Yeah, so these are the genes that are very, I mean, specialized in this, specialist in a way that they only being expressed in endothelial cells. And that's how I identified, I labeled them as specialist genes. Generalist genes are the ones that are just basically do everything and they exist in all cell types and just very, you know,
What
normal.
are what are endothelial cells?
Oh, endothelial cells are the vascular cells, cells that are inside the vasculature. They're the barrier between blood and every other tissue, like inside, lining inside the blood vessels.
Okay, so these are
Um,
just endothelial special cells. Okay.
yeah.
Good. Then let's continue.
Let's skip here. Like just focusing on endothelial cell, something that we found which was interesting was the genes that being expressed in the endothelial cell can have what we call polyfunctional characteristic, and not only endothelial cells, on most of the cell types. But just looking at the von Willebrand factor, which is a protein that being expressed during blood coagulation and plays a very important role in creating this platelet activation and creating, you know, fibrin structures to prevent bleeding. That endothelial cells basically, so these are endothelial cells and they express VWF. And when there is some type of, you know, injury, they interact with platelets and create like this huge, massive things that tries to do regeneration here and prevents bleeding. Just looking at VWF gene, like across different phenotypes, we identified like two major clusters of VWF. And these are the clusters that just we identified based on embeddings of VWF. So we just created a lot of endothelial cells in the model and got the embeddings for VWF on the output. And when you look at it for different sex, for different ages, for different tissues or any cell type, it doesn't really correlate with any of those things. But interestingly, when you look at the interactions of VWF with other genes in these two clusters, they call cluster 0 and cluster 1, you can see that it creates a different network in these two clusters. This one, the cluster 1, is the one that's like more canonical to VWF that we know. It's like involving blood coagulation, hemostasis, and wound healing. It's mostly working with other immune-related genes to prevent bleeding. But this other one is like less known, and I couldn't find anything about VWF interaction with these genes, especially like CLDN9, 5, and selecting P. Like these are more involved in the reactive oxygen species, and also glucocorticoids. So this is not, we still don't know why we have like two clusters of VWF here and why this the model creates two different token types for, not token types, two different types of embeddings for VWF, but this like kind of tell us that maybe there is something more about like this function of the genes that we didn't know of, right? Like we traditionally think that one gene being expressed only in conditions that do a specific thing, but maybe that is not the case, and genes play different roles based on the context that they are at. And this is not only the case for VWF, where we showed it for CD55, especially a lot of extracellular genes have this characteristic that they play multiple roles. And this is not the case, for example, for intracellular genes. This is KLF5 gene that basically you don't see that much that clustering, any type of clustering here. I'm rushing through this data, but just focusing on age again, I think something that we found really interesting was that, you know, different tissues age differently in this process. So there are certain tissues that have, when you just characterize like tissue types, phenotype based on the age that we had, you can see that some genes, some tissues are more similar than the other ones in the process of aging. And this is also kind of interesting because we know that a lot of tissues have different rates of aging, and that play a role in longevity research and basically targeting life extension pathways. I hope that I kind of convinced you that this maybe it's interesting to look at integrated genetic as opposed to reverse and forward genetics, and this is maybe a new method of like looking at this kind of genotype phenotype relationship. We're hoping to explore some of the results that we got here and do more validation of this result. Okay, with that, I would like to thank Johannes for giving me this opportunity and like open to any questions or anything. Oh my God, we have 16 messages in chat. I didn't see this, but I'm open to any questions or anything we can discuss, or maybe like next steps or ideas that you have that we can improve this work.
Okay, yeah, then thanks for the presentation. And then a little question for me first. How do you split, or how do people commonly split data in this realm when you, for example, do your computational evaluations?
What do you mean split? Yeah.
Like, how do you, what data do you train on and what data do you test on?
Oh, so we use the same. So we got the tabular sapiens, which has been balanced for like a lot of these phenotypes, right? It's an effort from Chen Zuckerberg Foundation that they try to get donors that are healthy, and they got different tissues and try to balance between sex and age and tissue types and cell types. And it's just we got that data set, which is 500,000. We shuffled all of it, got some of it for training and some of it for testing for this work. So that's how
So
we
this is a
basically
random split.
Yeah, random splitting.
Okay, and is there something different that people usually do in this field?
Not really. I don't. So usually you don't, when we train like data sets, like I'm just trying to think about like other methods that have done this kind of work. Transgene-former and single cell GPT, they basically trained on a bunch of data from different sources, like 30 million, 15 million or something. And they fitted like new data, and they just ask it, okay, do clustering on this ones. And the way they do it is usually they test it on some data set and train it on some data set, test it on different data sets. That's what I've seen before.
Okay.
I don't know how you guys do it. Like,
I don't deal with it. I've never used data like this in anything.
okay, but in your data, how do you?
Yeah, for example, if we have some task for small molecules, then oftentimes it's the case that you can tell the property of one molecule. Like if you have a similar scaffold, like the core of the molecule is very similar to the core of the molecule of another, and the molecule just changes with like an extra metal group stuck on somewhere. And then basically a chemist could already tell you that the properties of these two molecules will be very similar. Or it's very obvious that these properties are already very similar. And then thus we have like a scaffold split where we check that in the training and test set, you always have different scaffolds. Or in when we do something like docking, or like let's say anything related to proteins, you can always do a sequence similarity split and can check that you can generalize to proteins that are also a little bit different and not just the protein where only one amino acid is changed. And now maybe could do like an expression data split or something where whenever the similarity of expression data is above a certain threshold, then you make sure that the stuff is in the either in the training or in the test set, and you never have something that's above a certain similarity, or something in training set is never has more similarity than 30% to something in the test data.
Yeah. Yeah, we randomly sampled the data set to make sure that we have everything, but like we shuffled everything, and this data set has been balanced for a lot of different things. That's why we used it, and we try to do it on a very small like model data set like this because we want to make sure that we're not introducing any type of bias about any type of phenotype here.
Yeah. Okay. Oh yeah. Someone asked, can you show the data set statistics? Do you have a slide like that?
Yeah, I don't have it here, but yeah, it's in the paper. We published it, and I can reference the paper that basically published this data set here. It was published in Science like last year, a year and a half ago. If you search tabular sapiens, you'll find it. 14 donors basically for
Okay.
different tissues and cell types.
How many individuals data was used?
14 donors. And what are you? Yeah, so I think one of the interesting things.
So all the data that you have is only from 14 people.
Yeah.
Huh. I thought, yeah, see,
And
I don't know this field at all. That's surprising to me.
yeah, and where the next step that we're taking basically is to put more like create a better model, like the Chen Zuckerberg Foundation now has around 83 million data points. So we're trying to train it on all of those data of those to see what kind of, because that one has also the diseases, as a lot of different, tissue types from so many more patients. So the hope is that we can generalize like actually like getting more data for specifically disease. I think I didn't report anything. We didn't do anything.
What does data
Disease.
point here mean? Like, what is one data point?
Cell on cell.
Oh.
Yeah.
And so how many data points do you have in your 14 people data set?
500,000. Okay.
Okay. Well then, how are those collected?
Yeah, so this is a very clean data set, at least tabular sapiens, because they use the same sequencing machine, same procedure. They remove the batch effect with their like experimentally and computationally.
Oh, I just mean very
So
naively. Do I go ahead and poke the person once, or do I poke the person 100 times over 100 days or something like that?
no, they get biopsies from patients, from different tissues basically.
Okay.
And the ones that exist like now public, they are usually from diseased cases, like there is a biopsy from, whatever liver that had some type of disease and they got samples from it.
Okay.
Or it's blood, they
Then
get blood or yeah.
how does your approach compare to single cell GPT?
Yeah, single cell GPTs, I think, they are there are certain things that are similar, we're both using language models, I think. That's the similarity. Differences are we're looking at, I think traditionally people in the single cell RNA are still obsessed with, clustering methods. They're just the only task that they want to define is, okay, I have these gene expressions. How can I get cell type annotations? They give you like they train on a lot of data for the model to understand how can cluster different tissue cell types from each other. But I think we are integrating more data to the single cell basically. That's one of the innovations that we had, and adding sex and age and a bunch of other things that we're trying to add to single cell. We're trying to make it more comprehensive and kind of help the model to understand more about the biology here, not only the gene relationships, but also the gene relationships with the phenotypes that exist. And you can imagine that would be really useful in a lot of contexts. Also from method part, they also use BERT, but one of the challenges that we have seen in the single cell field is that people use language models, but they assign, you know, position to genes, and that's what I think mentioned. That is basically wrong. Doing that, it's not wrong. It's just like it maybe reduces the accuracy, or you can change your loss function probably if you do that. But here position is important, so we made sure that we have each gene in a specific position. That's something else that's different. They trained on a lot of perturbation stuff. We didn't really have perturbation. For me, at least personally, perturbation is not really important. I don't care about it. I know biologists are very obsessed with perturbation analysis. I don't think that's very important. Yeah, I think we look at single cell data in very different ways.
Okay. Well, then I would, oh, is there another question? Is the claim that the model naively learns interactions based on expression data? Do the outputs change if you reorder the gene's positions? No, I mean, the outputs are deterministic, like the reordering the gene's positions will not change the output at all. Is that right?
Yeah, reordering doesn't really, I mean, in this case, it doesn't really change anything because, and
I mean,
we
the
want
model
that,
is
right?
still doing exactly the same computations as if you had the other
Exactly.
order.
Yeah, exactly. But it's important
That's
to keep the same order for any type of, you know, training that you're doing. If you change that order, like basically your model is learning one thing with this data point and like another thing for another data point.
wait,
And it's just,
I thought the order was permutation invariant and you have no positional encoding for your genes.
yeah, we have positional encoding, but we keep the same position for each gene. So each gene,
Oh, okay.
yeah.
I see.
Whereas you can do the opposite, like, right? Like that's what single cell GPT is doing, that they define a vocabulary for gene IDs, and they every time like it just goes to a different position each gene, which is similar to language. But that's not true. That's not really correct, because the gene can be very first of the sentence, cell sentence, or the last. It doesn't really matter. But in language, it matters where the words are.
Wait a second. Yeah, someone also is raising a very good point in the chat.
Mhm.
Like, right, you randomly shuffle your data. This means that your model was trained on the data of every patient, like of all the 14 donors.
Mhm.
And then also means that yeah, you've trained on, like, right, if you now predict the age of these 14 age values of the people, then you've already seen someone with that same age, or you've already seen some data of that person with that age, and like it's exactly
Yeah,
the
but
same person. Yeah.
yeah, that is true. That is true. But you know, you have when you, the hope is that, I mean, it's not the age, it's not the only factor, right? Because you have age and tissue and cell type and sex. I mean, let's say sex is okay, there's just there less variation there. But age for one tissue is different from age for another tissue. You know, even though that they have the same number, but based on gene expression data, uh, tissues have different aging process. So you can't really say, okay, if you just look at the phenotypes, yes, it makes sense. People have seen one patient, another time seen the patient, but the genes that it's seeing are totally different. So now it needs to understand, okay, I saw the same age, but the expressions are very different. Now what's going on? So it is true that the data set is small and we don't have like that much We can't really generalize a lot of these things, but I'm pretty sure if you, because we have a lot of phenotypes, and if you even increase that more, we
Yeah,
might
so
decrease
with
some.
what were the 5 phenotypes again?
Sex, age, tissue, and cell type.
4. Okay.
Yeah.
Thank you. Yeah, okay. But then why don't you do something like a person split? Like that seems the most reasonable to me. Like you train on 10
One
people and you
person.
evaluate on 4 people.
Yeah, that is something they want to do, but here, because we have some samples from more, some samples are more from some patients, and some samples are less from some patients, you know. That's also another way of splitting. Yeah, I agree with that. That's possible.
And that's probably the way that, or I don't know, but I would assume that's the way that one cares about in the end, right? You train on a bunch of people and then
Hmm.
you have a person and you want to predict some stuff for them from what you were able to
Yeah,
experiment together.
if you want to predict the age of a person, that is the right way of doing it. But if you want to predict
But
the age of a
no, I
cell,
never want to predict the age of a patient.
but if you want to predict, for example, age or sex of a cell, that's a harder task. Like, it doesn't matter like what patient is coming from. It's important what kind of gene combinations you have there.
Yeah, okay. Good, good. But then I think we can, yeah, I want to come to an end and thank you
Mhm.
one last time. And then, yeah, any last points from your
Yeah.
side of the day?
No, thanks for the time. I know that like this is very the like, you know, it's the emphasis of like this kind of work. If anyone has like, you know, ideas and, you know, anything that we might, you know, I talked with Johannes to, you know, integrate more kind of data modalities to it. I think we have some potential to, you know, use this kind of models for understanding or predicting, you know, aging processes, gene interaction, side effect processes. So these are the things that we're thinking. If you guys have any idea, or you think that there are better ways of doing this, I'll be really happy to chat about collaborations, or just, you know, brainstorming.